Understanding meiosis, part 1, chromosome structure. It occurred to me when I was thinking about how to explain meiosis better that I think there is often a fundamental misunderstanding about chromosome structure at the root of people's problems with meiosis. So I thought I'd make this video to try and address that problem before going on in part 2 and 3 to look at the mechanism of meiosis and then how meiosis leads to variation. So let's start off with the basic structure of a chromosome. So here we have our basic chromosome, and our basic chromosome is made up of chromatids, and it's important to remember that the chromatids are completely identical to each other. They are a copy of each other, and those two chromatids are held together by a centromere. Okay, and that is a single chromosome. In a diploid cell, chromosomes hang around in homologous pairs, and in these homologous pairs, what we have are the same genes but different alleles. So this is one chromosome in a homologous pair that is made up of two identical sister chromatids. This is another chromosome in the homologous pair made up of two identical sister chromatids. But this chromatid and this chromatid, they will have the same genes at the same gene loci. So here and here, they will have the same genes, but they may have different alleles of those genes. Not necessarily, they could be the same. If they were homozygous, if it was a homozygous organism, the alleles would be the same for that gene. But if the organism was heterozygous, then obviously the alleles would be different. So that's a homologous pair of chromosomes. Same gene, different alleles, and that's what we see in diploid cells. So the big question really is what happens in interphase, because this is, I think, the key for really trying to understand what's going on. So in mitosis, what happens is that the individual chromosomes are separated into their chromatids. So we have prophase, metaphase, anaphase, telophase, and in doing that we go from a complete chromosome here to individual chromatids here and here. That cell will now, or these two cells will now undergo interphase, and in interphase, during the S phase, the DNA replicates, and so these individual chromatids now become complete chromosomes. Now, if we apply this to meiosis, in meiosis 1, we have our homologous pairs of chromosomes separating, and in this at this point, we do not undergo interphase. If one of these cells did go through interphase at this point, then the complete chromosome would be replicated. So what you would find is you would have another complete chromosome here, which obviously then returns the cell to being diploid and completely negates the whole purpose of meiosis, which is there's a reduction division, which is why in meiosis we go I, P more after T, more P more after T, I. We don't have an interphase in the middle. So in meiosis 2, what happens is that these complete chromosomes then split and become the individual chromatids. Now at this point, these 4 individual cells, so if you think about it, we have 1, 2, 3, 4 haploid cells. They do go through interphase, and consequently they do then replicate their DNA. So this will now become a complete chromosome. This will now become a complete chromosome. This will now become a complete chromosome, and this will now become a complete chromosome. And that's how we have haploid cells with, in the case of humans, 23 single chromosomes from a diploid cell of 23 pairs of chromosomes. Hopefully that will help you to start to get your head round how we go from diploid to haploid, because I understand that seeing that you think, well, how is that a haploid cell? That doesn't make any sense. It's just a single chromatid. I think one of the things just to be aware of with anything to do with meiosis is that we can be quite lazy when we talk about meiosis, or chromosomes in particular. And whilst sometimes you see chromosomes represented like this, you will also see chromosomes just represented as essentially as a chromatid without the centromere in the second chromatid. The reason, of course, why that can be the case is simply because you literally have, one chromatid is exactly the same as the other, and so it can be a kind of a lazy shorthand. Try not to get too worried about counting kind of 46, 92, 23 and so on. Just stick to the basics and really understand how we go from chromatid to complete chromosome in interphase after the second meiotic division. I hope that helps. If not, come and see me.